So coming to my talk, as I said, I will talk about 24-plex. This is QIAGEN's new forensic STR kit that allows global STR analysis, including the quality sensor. Okay, in the next 40 minutes, I will present you, of course, the content of the kit, as well as the kit features and the benefits. During the early development phase, we thought about what are the issues customers have to deal with in the routine analysis and what complicates their informative profiling success. Some of these questions I have listed here. So, do customers have sometimes issues with marker overlaps? So, for example, the amplicon of a certain marker is of so large fragment size that the peak appears in the next upper STR channel. Is this a signal or noise? So this question deals with the sensitivity. On the one hand, of course, signal sensitivity is due to signal peak height, but is also related to signal to noise ratio to clearly determine an allele. So if you might have a high background in the past, you might have lost some peaks. Another question, is it really a male profile? So maybe you lost a melogenin peak in the past. Do you have to rerun samples? How do you know that the PCR failed, or is it a degraded or inhibited sample? And are your results international compatible? And does it include, for example, the European marker SE33? In the next 40 minutes, I will give you answers on all these questions. We at QIAGEN work very hard on these topics to bring STR analysis to the next generation. So this is the agenda for today's webinar. First of all, I will give you an overview about the kit concept. The Investigator 24-plex QS and the Investigator 24-plex Go kit. So actually, this is a two-kit format, as you can see. These two kits are very similar, but are developed for different applications. The next topic will be the innovative quality sensor. Then I will talk about the confirmed profiling success, and the last point is the availability of the kit. So first topic, the kit concept and overview. As I mentioned before, Investigator 24-plex is not only one kit, it is a family of two very similar STR kits. So on the left side, I displayed the Investigator 24-plex QS. This is intended to use for purified DNA from casework or reference samples. So if you have very difficult samples, you should use the 24-plex QS kit. On the right side, I displayed 24-plex Go. This is intended to use for reference samples, for example, from FDA cards or swabs. And this is a very fast and cost-efficient approach. Both kits share very similar features. I displayed some of these topics here in this slide. First of all, we address 23 markers that are recommended by the CODIS core working group. These 23 markers enable the international compatibility and will facilitate the data exchange between different countries. We included an innovative quality sensor. I will go into detail later on. We included SE33 and DYS391. So SE33 is a really high discriminative marker, and it's getting more and more popular and utilized in different countries around the world. Therefore, we decided to include this marker, SE33, and DYS391 is an additional gender marker. Both kits are based on a 6-color setup. So we decided to use 6 colors, one additional color to have a higher marker range available and to minimize marker overlaps between different markers to facilitate the data interpretation. And also to facilitate the data interpretation, we added 60 additional alleles to the allelic ladder. And one important point is new reaction chemistry. This is FRM 2.0. Here we have a new formulation that gives you an even higher convenience and stability. It is a very fast reaction. You can cycle, for example, a 24-plex QS in 60 minutes. And if you're cycling 24-plex Go for direct amplification, you are in the range between 45 and 50 minutes. This depends on the cycle number you use. We have here a very high inhibitor tolerance, so this is still the same as known for the FRM with the plus chemistry kits. So these kits have the highest inhibitor tolerance on the market right now, and the FRM 2.0 is on the same level with a very high inhibitor tolerance. We have a very convenient pipetting. We see FRM 2.0. Some customers reported when using the FRM of the first generation that they had sometimes issues with viscosity, or they had bubbles in their reactions, and this was due to development reasons. So we wanted to get a very high inhibitor tolerance and very fast speed, and this had the disadvantage of some bit viscosity. But now in the FRM 2.0, we have a much better viscosity, so it is more fluent. You don't need a shaker. It's very easy pipetting, and you still have this high inhibitor tolerance and this high speed in combination with the lower viscosity. So we are very happy that we could develop this new fast reaction chemistry. Okay, in the next slide, I present you the markers that we introduced in the kit. So first of all, if you look into the history, you can see in this table on the left side are the CODIS markers that are predominantly used in US or other countries around the world. And on the right side, we have the European marker set. And the CODIS core working group, they recommended to use all of these markers. So this is a combination of the CODIS and the European standard set. And with 24-plex, we will include all of these markers. So we included SE33. So as I said before, we think this is a very good marker. It's highly discriminative, and it is getting more and more popular and utilized from different customers around the world. So we think this is a good choice to include the SE33 marker. We have also the gender marker, DYS391. So DYS391 and amylogenins, these are both used for gender identification. And with this, you have all required CODIS expansion markers in one kit. Okay, next slide. I present you the new colors. We set up 6 color matrix, so it's called BT6. I hope you can see it in the upper figure. So we have a new color. This is a purple color. This is shown here. The wavelength maxima. This is between the orange and the red maxima. And we were happy to find a new color that fits to the other colors with a very high intensity, and with this you get also a very high sensitivity. And this is also a good choice, as we had really low pull-ups between the different channels. On the lower figure, you can see the electrophoretic separation of the different fragments from the matrix. So it's starting with orange, then red, yellow, green, blue, and purple. So these are all separated by the matrix installation, and the baseline is very flat. So this would be very easy for you to install the matrix, and you will get no issues regarding matrix installation, and you will get really low pull-ups. Okay, next slide shows you the marker arrangement of the 24-plex kit. So this is a schematic overview. This is comparable to your electropherogram. So you see markers in blue, green, yellow, red, and purple. And this displays the marker range and that the peaks of a certain marker can occur. In the development, we looked for primers that were already existing in our kits. So we had one block of primers that we took from ESS Plex SE. Then the next row we took from ID Plex Plus and coupled the primers with a new 6 color. And we had to design 3 new primer pairs. So on the one hand, this is DYA391. So this is not present in ESS Plex SE or ID Plex. We designed new primers for SE33. This had the reason to fix discordant results that were shown up in ESS Plex SE. And the third primer is for D7 because otherwise there was no space available for the D7 marker. So we looked in the development for primer arrangement and design in that way that we minimize the marker overlap between different markers. This will make it more easier for you to interpret the results. Okay, yeah, as all of these primers are trusted primers from existing kits that most of you know, this stands for reliable results. In this slide, I showed you the marker arrangement again. So we have the markers addressed in the different colors. So this is 6FAM, BTG, BTY, BTR2, and BTP. And as you can see out of this scheme is that we introduced the quality sensor system. This is QS1 at the front, and this is QS2 at the high molecular range. So these are 2 markers that are not STR markers that are used to verify the quality of the PCR. This I will point to you later. Here show you the allergic ladder of the 24-plex. In comparison to existing kits like ESS Plex, SE or ID Plex, allergic ladder of 24-plex will contain about 60 additional alleles that will facilitate the interpretation of allele calling. So what does it mean to the routine analyst? For alleles that appear in high frequency, but also for very rare alleles, the probability of automatic allele genotyping is very likely, and this will increase the genotyping efficiency, and it will make it easier for you to interpret the results. So you can see that there are lots of alleles present in the allelic ladder, and yeah, this should make it more easier to automatically genotype the alleles. Okay, after the kit overview, I will now come to the Investigator 24-plex QS in more detail. So Investigator 24-plex QS is intended to use for difficult samples. If you, for example, have purified DNA from casework samples, or you can also use it for reference samples. This preliminary electrophorom that you can see here is the result of the PCR done with 500 picogram template DNA of 9948. And what you can see that you have a very balanced profile throughout the colors and throughout the markers. And yeah, you can see here at the front, and this is QS1 sensor, and at the high molecular range, this is a QS2 quality sensor. Okay, next slide displays the baseline of the kit. So we performed no template control experiment with no DNA in the PCR. And what you can see is that you get a really flat baseline. So of course you still have the quality sensors QS1 and QS2. These are present in the PCR. So you have primers and templates that are included in the primer mix, and these peaks will be amplified in every PCR reaction. In that case, if the PCR was successful. But what I want to point out is that the baseline is very flat here, and this will make it very easy for you to interpret the results. So, for example, if you have casework samples with low template DNA, so it will be very easy for you if you, for example, have a threshold of 50 RFU. You can easily select a certain peak for allele genotyping, and you will have a very high sensitivity as you have a really good signal to noise ratio. Okay, next point is a 24-plex Go kit. So this is for direct amplification. And first of all, I would like to give you an overview about the workflow of the Go kit. So usually you can use different kinds of sample types. The first example is if you have blood on FDA cards or other paper. First of all, in the protocol, you have to take a 1.2 millimeter punch from the center of the blood spot. Then you have to prepare a PCR master mix. Then you transfer a disc to the PCR reaction, and then you start the thermal cycling. So this is 25 cycles here. This is about 45 minutes, so very fast cycling. And then you go to the capillary electrophoresis to analyze the peaks. So this workflow is very easy. So you have to prepare your PCR, throw in the punch of the FDA card, start cycling, and then after the CE analysis. The workflow for buccal cells on FDA cards is very similar to this. So here again, you take a punch from the center of the spot, you prepare the PCR master mix, you transfer the disc to the PCR reaction, start the thermal cycler. Here we recommend 26 cycles, and after this you start the CE instrument. Third option might be if you're using buccal swabs for reference samples. So here we have a very fast lysis protocol. We developed a special buffer. This is called STR Go lysis buffer. And with this buffer, you can perform the lysis in 5 minutes. This is the first step that you have to do. Then you prepare the PCR master mix. Afterwards, you transfer 2 microliters of the swab lysate to the PCR reaction. Then you start the thermal cycling, here 27 cycles, and then you start the CE. We recommend so cycles between 25 and 27 cycles. This is the cycling time between 45 and 50 minutes, so this is really fast. But this also depends on the type of sample that you are using, and it might be possible that you have to use 1 cycle more or 1 cycle less to assure a very good first round success rate. The Politis protocol looks like this. So you have to collect your sample, place it into a tube. Then you have to add 500 microliters of the STR Go lysis buffer. Then you have to incubate 5 minutes at room temperature, and that's all. Then you can transfer 2 microliters to the PCR and go for cycling. This lysis protocol takes only 5 minutes at room temperature. It's a very robust non-enzymatic lysis. You can use it for different kinds of swabs, cotton, nylon, or flocked swabs. And what's also a very nice feature is you can use it on the one hand directly to go directly forward to the PCR, or you can also store your swab lysis, for example, at minus 20 degrees. And you can use it for rerunning samples at a later time point. So QIAGEN's innovative STR Go lysis buffer reduces lysis time by at least 75%. So if you compare it, for example, to protocols that are routinely used or used from other suppliers. The next slide, I have an overview about one example of the experiment with buccal cells on swabs. So here we did a lysis in 5 minutes, transferred 2 microliters to the PCR, and the result is what you can see in this figure here. So you get a really nice balanced profile. Starting from beginning to the end is a really balanced peaks, and it's very easy interpretation and analysis to get the genotype of this sample. The next point is if you use another sample type. This is blood on FDA paper. Here we used a 1.2 millimeter punch, transferred it to the PCR reaction, and this is a result that you can see here. So this is also again a really nice balanced profile. What is interesting in this case is this is a female profile, so you get only 1 peak for amylogenin, and you don't get any peak for DYS391. So DYS391 is a marker that is located on the Y chromosome, and therefore you have verified that this is a female sample here. What's interesting for reference samples that he has most very high number of samples that have to be processed, and for this we had a cooperation with Hamilton to facilitate the handling of the PCR reaction setups, and here is an instrument called Hamilton Easy Punch Starlet, and we validated it for the 24-plex Go protocol, as well as we did also for other Go kits that QIAGEN is selling. And this is very easy setup. So as you can see in this figure, first of all, the instrument pipettes the master mix in each well. Then the roboter starts punching the spots directly into a well, and you have to program the instrument, and then you can go away, and it usually takes 90 minutes for 96 samples. 90 minutes means this is a long time that the PCR reaction stands at room temperature, and here primer-dimer reactions or unspecific reaction might occur. But we developed our new FRM 2.0 chemistry in that way that this is very stable. So we have a very tight hot start function, and you can also use more than one well. You can, for example, if it takes 4 or 6 hours, it doesn't matter, because the hot start function is so tight that no primer, dimer, or unspecific reaction occur, and you have a very clear profile. So this Hamilton Easy Punch is a true walkaway automation for optimal results, and you can use it for FDA paper with the F24 Plex Go kit. Okay, next point. This is one of the highlights of this talk. This is the innovative quality sensor. So I grabbed one question that I raised at the beginning. So do I have to rerun samples? Is it degradation or is it inhibition? And the answer on this is the quality sensor. So actually the quality sensor consists of two positive controls in the purple channel, as I've shown you before. This means you have certain primers and the template that are already included in the primer mix. So you don't have to do any other handling steps. So you just set up your PCR as usual, and as a benefit you will get the results of the quality sensor. The template is an artificial template sequence. This matches to does not matches to any other known sequence known so far. So you will not get in trouble with any unspecific peaks. The quality sensor will distinguish successful amplification, degraded DNA, inhibited DNA, no DNA, or if the PCR reaction failed. I will explain it in the next slide to you how the quality sensor system works. First of all, if you have a standard sample, so this is not degraded, this is not inhibited, you see the STR peaks that are amplified as usual, and what you also can see is that you have quality sensor 1 in the front and quality sensor 2 in the high molecular range that are both amplified well. So this is how a standard reaction looks like, and it confirms that your sample is not degraded or inhibited. If you perform a negative control, for example, or you have a casework sample where you don't have any DNA in your sample, then the profile would like this, as you can see here in this figure. So you can't see any STR peaks because you don't have any DNA in your sample, but quality sensor 1 and quality sensor 2 are present. So these are present because you already have your template. This is the specific template from the quality sensor. This is in every experiment present. So we always will get the peaks from the two quality sensors. And so this shows you that there was no DNA in the sample, but the PCR worked. And in case of you're doing casework samples, you don't have to repeat your experiments because it will not lead to better results. Or another case would be you can increase the volume of your template DNA to get maybe any small peaks above the threshold. Okay, next scenario is if the PCR reaction failed, then you see an electrophoreologram like this. So you don't get any STR profile, and you also don't get any quality sensor 1 and 2, so they're both missing. You have totally flat baseline, so there's no peak present, and this shows you that something went wrong in your PCR. So maybe you forgot the primers or the PCR buffer or to do cycle or whatever. And this shows you that there was that the PCR reaction failed, and a rerun will most likely lead to better results. So now it's getting more tricky. You have here one profile that shows you the so-called ski slope effect. So meaning you have high peaks at the front, and you have very low peaks at the higher molecular range. And if you have an inhibited sample, then the difference is that you have a high quality sensor 1 in the mini STR region, and you have drop down or drop out of the quality sensor 2. So this shows you that the PCR is inhibited and doesn't work well. And you can rerun the PCR, for example, if you try to dilute the sample, or maybe it's also a good choice to purify the DNA again with an appropriate kit. Okay, quality sensor, here's one example for an inhibited sample. So we spiked 750 micromolar hematine into the PCR, and this is a typical electrophoreal profile that you can see here. So the peaks are going down from the beginning to the end, so you have low peaks on the STR regions. You also can see some split peaks. And you have a quality sensor 1 that is working fine, and the quality sensor 2 that is missing here in this case. Next scenario is a degraded sample. So this is comparable to inhibited sample, but the difference is that the quality sensor 2 is still present. So you have quality sensor 1 and quality sensor 2 that are both amplified well, and you have this ski slope effect. So you have high peaks at the mini STR range and low peaks at the high molecular range. And this shows you that the PCR works perfectly, but there's an issue with your sample that is degraded. And as you don't have any template of high fragment size, it is not possible to amplify these peaks, for example, in the D7 marker. So a rerun will not improve here your profile, and 24-plex already gives you the best results as a standard setup. You again have one example for you. So on the left side, you can see an agarose gel. The first line is the control of genomic DNA, and we shared it by ultrasound to different fragment sizes, so to 500, to 300, and to 150 base pairs. And you can see this fragment range on the agarose gel, and we placed we used this sample of the 150 base pair shared fragments in the PCR, and the result is that's what you can see on the right side. You can see that you don't get fragments of the higher molecular range, and therefore you have good amplification of in the mini STR regions, low amplification rate and the higher molecular range. And what's really nice to see is that the quality sensor 1 and 2, these are both present and amplified very well. So in summary, the benefits of the quality sensors are that it enables successful amplification. So it enables you to distinguish between successful amplification, a degraded DNA, an inhibited DNA, if you have no DNA in your sample, or the PCR reaction failed. And this is for short summary. So a standard sample, the quality sensors and the STR markers are amplified very good. If you have a negative control or no DNA, then you don't have STR markers, but both quality sensors 1 and 2 are present. If you have issues with your PCR, for example, if the PCR reaction fails, then you don't have STR markers and you don't have quality sensors. if you have an inhibited sample, then you see the ski slope profile with high peak heights at the beginning and low peak heights and the high molecular range. So this is identical between inhibited sample and degraded sample, but the difference is that with inhibited sample you don't have, or you have a drop down with the quality sensor 2, and for the degraded sample you still have the quality sensor 2 present. Okay, next topic is confirmed profiling success. First of all, I will introduce you in the gender typing with 24-plex. So 24-plex includes 2 gender markers. This is amylogenin and DYS391. And yeah, in the past, some population studies showed that there are some mutation sites on the Y chromosome that leads to failed typing of amylogenin. So there are some studies from India and Malaysia that showed that there are, for example, point mutation sites in the primer binding sites. This leads to drop down or drop out of the peaks. Luckily, the QIAGEN kit was not affected yet by this kind of mutation, but there are other mutations known that the whole amylogenin gene is deleted. And this is also not amylogenin, also the neighboring genes are deleted. So with this, you can't design a primer for this kind of scenario, because if the DNA is not present, you can amplify the amylogenin gene. And for this reason, we introduced the new marker. This is DYS391, and we placed it at a small size for very robust typing, also for difficult samples. And we checked the performance of amylogenin DYS391 while testing amylogenin Y null mutants. Here I have one of example of difficult sample. So we decided to place DYS391 in the relatively short fragment range for 2 reasons. So in the exemplary electrophoregram shown here, you see the results of degraded DNA. So amylogenin and DYS were very well amplified in this scenario. But if you had placed DYS391 at the high molecular range, you would not see any result of the marker DYS391. And the second reason is why we choose DYS391. And the mini STR reason is because of bioinformatics. So we performed a sequence alignment for DYS391 between the Y and the X chromosome, and it turned out that the sequence difference for designing PCR fragments of higher fragment size are very critical. And therefore we selected primers that are amplified at a relatively small fragment size. So in my point of view, the gender identification is very important attribute for the police to identify a certain suspect. So therefore, in my opinion, it's very important to get the right results for gender identification. So this is not a scenario that is reported very often, that there are mutations in the amylogenin gene, but from time to time there are cases reported. And for this, the combination of amylogenin and DYS391 is a very good choice. And as we have it DYS391 in the mini STR region, you can also perform the gender typing with very difficult samples. Okay, next topic, concordance. So we know for some kits, from, for example, ESS Plex SE or IDE Plex, there were some disconcordances reported in a very few cases, but they are, can occur. And with 24 Plex, we fixed all these disconcordances. For example, yeah, if you look at SE33 marker, so before it occurred that you have a drop down or drop out for a certain sample, and with 24 Plex, now we have fixed this issue, and you get a very well amplified SE33 marker, and both peaks, so the heterozygous profile can be easily identified. So we have a very high concordance for reliable results, and this was also tested by a certain institute. So this is a NIST institute in US, and they performed a concordance study for us with the investigator 24 Plex QS kit. And the NIST institute compared a final data set with 656 unrelated individuals. So it's a population set of 650 samples and the SRM2091C samples. And it showed that we have a full concordance with this NIST SRM2391C certified values. We have no null alleles, but we had one disconcordance regarding the marker D7. It was one sample out of this 656 samples. And what was interesting is that this D7 24 Plex result is concordant, for example, to kits like Identifiler, Plex Fusion, Profiler Plus, but it is discordant to other kits like Plex 16, Minifiler, or ID Plex. Yeah, so out of this about 30,000 alleles compared, there was one discordant call. So this is 0.003% disconcordance, or the other way around, you have a 99.997% concordance between the investigator 24 Plex kit and the NIST final data set. So with this 24 Plex kit, so we have fixed all the disconcordances known so far, and you will get really reliable and very high quality results. Okay, the last part is the availability. So, as mentioned before, we have a two-kit format. So this is 24 Plex Go for direct amplification. These kits will be available in formats of 200 and 1000 reactions. And these contain, of course, primer mix, reaction mix, control DNA, alleelic ladder. And what's also present, included in the kit, is a DNA size standard 550. As a BTO marker, so you don't have to buy this separately. And the second kit, 24 Plex QS, this will be available in 100 reactions and in 400 reactions. And here you also have a primer mix, reaction mix, you have water to fill up the reaction volume, control DNA, the alleelic ladder, and again the DNA size standard. I forgot, so you also can, of course, buy the STR Go lysis buffer, yeah, under this number shown here. The launch of this kit is planned for September this year, 24 Plex QS, and 24 Plex Go will be launched in end of the year in November. Okay, the last slide is a summary. The investigator 24 Plex kit, this is a kit configuration for direct amplification and for the analysis of purified DNA. So we have a kit family of two kits. This is 24 Plex QS and 24 Plex Go. The kit includes all new CODIS core loci and the European standard set of loci. So we address all markers that are recommended by the CODIS core working group, and it makes it easier for you to exchange data between different countries. We included here the quality sensor system, and I hope I could explain you which benefits you will have when using the 24-plex kit, so you know much more information about your sample and how to process further in your workflow. We have a high concordance, so this is a very high quality kit with high concordance to reduce the risk of misinterpretation. We have a very high sensitivity with a superior peak balance, so the baseline is very flat, and it's very easy for you to call a certain allele. So there's a very nice signal-to-noise ratio, good peak heights, and a really good peak balance. Okay, with this, I like to close. I hope you enjoyed the webinar, and I hope I could answer all the questions that are raised at the beginning of my talk, and I'm very happy to answer your questions if you have any further questions.
QIAGEN.
Sample and assay technologies.